So this is where we left off in the last video, and we worked ourselves up to this concept of DNA. And we said DNA is made up a couple of different pieces, and it really depends on how far down we want to go to talk about DNA. So we said nucleosides basically are coming together and holding hands to make nucleotides, and then nucleotides are going to be hooked together, and that's going to make nucleic acid. And nucleic acid will kind of come in two forms. There's different versions of it, and one is a deoxy version that we call deoxyribonucleic acid, aka DNA, and then the other one is ribonucleic acid, which we abbreviate as RNA. Each one with its own purpose, each one with its own activity. So here's the deal. With us, when we're in a laboratory, we really like to look at things from the bottom up. That traditionally is how lab analysts are supposed to be, especially in the field of chemistry and chemistry-related fields, okay? So biologists typically look at things on a broader scale. They look at the bigger picture first, and then they worry about the small things later, even if they worry about the small things. But we're just the exact opposite, and that's always the constant battle that chemists and biologists have with each other. So with us, we are going to turn this DNA and we are going to work up. I think this is a better way to learn DNA and what it's doing over a course of time versus working down and trying to complete it with that model. So you are getting ready to learn about a chemist point of view when it concerns the DNA molecule. All right, so how are we going to start this off? Well, first, I told you in the last video that these things were made up of bases. And I had two choices, purine and pyrimidine. So these bases are going to be called heterocyclic bases. Hetero, of course, means different. Cyclic means a ring. So it is a ring structure. All right. 1, 2, 3, 4, 5, and 6. All right, there's part of it. Because DNA is very stable, RNA too, by the way, anything that leads to stability of these compounds are going to be present. And this is an alternating double, single, double, single, double, single all the way around. We call that delocalization. It's a conjugated ring system that is delocalized. Conjugated means double, single, double, single, double. Delocalization means that the electrons can now float in that area of the molecule, and they're not really locked into place. They have some movement, they have some freedom, and they enjoy that, just like you would. But these are called heterocyclic, meaning that at one of these carbons, it's not really a carbon any longer, it's something else. And in our case of the heterocyclic bases with DNA, they are nitrogens. So one of those carbons will get exchanged for a nitrogen. And this is not uncommon. When we talk about organic chemistry, it is very common for us to see nitrogen and oxygen and maybe sulfur that's involved in the molecules. Phosphorus can be involved in these molecules as well. No big deal. And they're called a base because, as the definition of a base states, that they should be able to accept a proton. Well, this nitrogen has 1, 2, 3 bonds, and to fulfill the octet, it will need a set of electrons right there. And there's our definition of a base. That site of electrons will be able to accept a proton, and if it does accept a proton, then that qualifies for the definition. So these are bases if we look at their chemistries. Just their straight out chemistry the way they're written right now. Now, this particular setup, a 6-membered ring with a nitrogen and a free set of electrons, this is our pyridine model. Pyridine is going to be a reagent that you will probably talk about in organic chemistry. If you haven't talked about it up to this point, you will see it eventually later down the road. They will typically throw in pyridine when we start talking about halogenating an alcohol. You'll often see pyridine throw out quite a bit. But pyridine is going to be embedded into our DNA structure. And we're going to have to figure out how pyridine plays a role. But this is not the only option, right? I told you there were actually two options, right? And those two options I said was pyridine and purine. Well, how does purine look? Okay, so let's start off with our six-membered ring, like we've drawn up there. But off of this six-membered ring, we will have a five-membered ring that will come out. So almost like a cyclohexane, 6, and a cyclopentane, 5. Well, these are heterocyclic. So that also means that some of those carbons have been replaced by a nitrogen. So I'm going to erase this one, and I'm going to erase this one, and I'm erasing this one, and I'm erasing that one. There's quite a few more nitrogens in this. Okay, so I'm going to draw my nitrogen at this spot, and my nitrogen here, and my nitrogen there, and my nitrogen here. 4 of them. As with up here, we still get delocalization in this ring. It is still present. It is still there. I never really want to forget that. That helps stabilize this ring, or half of the ring at least, so far. And then on the other side of this molecule, it continues on actually. Double, single, double, single, and we wish that we could put a double here, but we can't. That carbon's overloaded at this point. It already has 4 bonds, so there's no more room for that. If I look at these nitrogens, they all have free pairs. 1, 2, 3 bonds, so therefore that needs a pair. This one, 1, 2, 3, and at the bottom, there's our 4th. The nitrogen on the top right-hand side, 1, 2, 3, and then here is the 4th. However, this one, this nitrogen, look, it only has 1 bond to the left and 1 bond to the right. It doesn't have a double bond like those other ones do. So there's got to be something else extra here, and there is. There is a hydrogen. That makes the 3rd bond, but you know what? It still has a free pair because that's what it needs to satisfy the octet. So 3 bonds in total. Hydrogen's not normally drawn. It's understood to be there, but I've drawn it just for reference. It still has a free pair. And this is what we call our purines. So DNA is made up of a series of pyridine and purines. Hold on a minute. Let me rewrite this word. You're going to kill me. I keep calling it pyridine. Pyrimidine. Pyrimidine. There we go. Pyrimidine. That's a little better. You should have caught me. You should have told me. Why weren't you screaming at me at the computer screen? Well, you probably are now due to that mistake. So pyrimidine and purines. That is what we're after. Okay. So with that said, here, magically, ta-da! We have our purines that we would like to talk about. And these purines are going to be located in DNA and RNA both. And there's only 2 options that show up in either one of these routes. That's it, folks. 2. 2 purines. If you look at the base of the structure, they're both very similar, right? It is really pretty much the template that we've talked about, but we've modified the template a little bit to make this work. You know, if everything was the same, then there would be no differences at all in the DNA, but there is. There's slight modifications to the purine and the pyrimidine bases. So what are some of these modifications? Well, adenine is to the left. So this is abbreviated with an A in the DNA world. And guanine is to the right, and this is abbreviated with a G in the DNA world. Both of these could also show up in RNA if they need to. No big deal. And if I look at the differences between these, first, it looks like adenine has a NH2. There's an amine right up here at the very top. That is called, that's what that functional group is. Well, we still have an amine here, but the amine is actually located in a different spot in guanine. So an amine is still present, no big deal. We've just shifted it a little. But guanine brings with it an extra functional group, and that extra functional group is right here. And that functional group is actually an amide. And the reason is because this is a carbon that's double bonded to an O that is now bonded on a nitrogen. And that entire functional group is an amide. If we're only focused on the C double bond O, that's a carbonyl. And a C double bond O within a longer ring is called a ketone. But in particular, if it's connected to a nitrogen on one side, like it is here, this is called an amide. So adenine has an amine to the left, and guanine has an amine as well, but it also has an amide. That's the only difference between the A and the G. So now you can kind of see why people were scratching their head many years ago. You're telling me that DNA, the genetic carrier, carries the genetics and all the information about someone. And here are 2 pieces of it out of 4. 1/2. And the only difference is the conversion of an amine and an amide? No, proteins are probably the way to go. Proteins are probably the choices that I need to do that carries genetic information. There's more options, there's more possibilities, it can wrap itself around in different ways, versus something like this. Little do they know, though, that idea is going to be blasted out of the water in the mid-1950s. Right? We've already mentioned it. All right, so there we go. Now let's take a look at pyrimidines. Pyrimidines do have a modification here, and there's 3 different versions. Cytosine is to the left, so here we go. And then thymine, which is the T, is in the middle, and then uracil is over here to the right. Notice they are modified yet again, and if we look at the base of the ring structure, the pyrimidine is going to add an extra nitrogen into the ring. That's what we're going to show up here, all right? This extra nitrogen, no big deal, it's in all of them. The 6-membered ring is in all of them. This other nitrogen is in all of them. And this double bond O is in all of them. Look at that. Double bond O, double bond O, double bond O. So what are the differences? Well, of course, this one has another amine group. And if we look at this one, this is a C double bond O on a nitrogen. And this is our amide functional group again. And we also have with it a methyl group. That's understood to be a CH3 right there. CH3. And then over on the right-hand side, we see the C double bond O with the nitrogen again. So there is our amide functional group. That's really it. Those are our 3 choices. You either get an amine, or you get an amide, or you get an amide with a methyl group. This is the other half of the picture. So the first half, they were really only different between the amine and amide. And in the second half, they're only different in amine and amide, and the answer is yeah. That's it. And these differences, people, really distinguishes your DNA from everybody else's. Not too many choices there, right? Well, let me make it even more outstanding for you. Because if I take a look at the right-hand structure, uracil, this only shows up in RNA. This is not in DNA, which is your genetic carrier. It's RNA only. So these four, that's it. The two to the left on the pyrimidine side, and here are your two purines. They're all rings. They're all bases, which are nitrogen-based, and they all play off of the amine and amide functional group. And this is what gives us a different genetic code. It's kind of crazy when you really stop and think about it. So purines and pyrimidines are very picky. When they come in proximity to each other, they're going to have a personal preference on who they would like to hang out with. All right, how to pick their partners. They're very particular when this begins to happen. The adenine, or the A, will always pair up with T or U. Remember, U is only used in RNA. T is what we'll find in DNA. So therefore, I can kind of leave out the U description right now because we're only really focused on DNA and that's it. So A pairs up with T every single time, and T pairs up with A every single time. There's no exceptions here. And the G pairs up with the C. The way that I remembered this in biology, I was taking chemistry at the time, and I was learning about pieces of equipment and organic and stuff at the time. So GC is an instrument that we often use in a laboratory, and that's the way that I knew that those two went together. GC, gas chromatograph. Well, GC also works out here too in DNA. It's meant to be. So anytime I see G, C will pair it with it. Anytime I see C, G will pair it with it. And that's it. Those are the two bondings. Now let's think about this. We have 4 bases. That's it. They're only slightly different. No major differences in them at all. A couple of nitrogens maybe, a couple of extra bonds, but that's it. And now those 4 bases are going to make 2 relationships. That's all. It's either AT or it's GC. So now let's redefine this a little bit. Your genetic code is only different based on these 2 relationships? No, this can't be. And again, this is why people scratched their head back in the day. There's no way. There's no way. 2 relationships, these 2 pairings are going to determine your hair color and your eye color and your blood type and everything else that will come along with you as a person, as an individual. How? How is this even possible? Well, that we'll get into. So before we do that, I want to talk about this pairing. Why does the pairing happen the way that it does? Well, it's all based on hydrogen bonds. So here are the 2 different relationships that we've just discussed, A to T's and G's to C's, and they are drawn out for you. Only the base pairs is what we're looking at. And what I want you to look yourself with, or what I want you to look at, is the dotted lines that happen here. H attached to an N, O, or F that is then attached to another N, O, or F. That is the definition of a hydrogen bond. Hydrogen bonds are the driving force that keep these base pairs together. In the case of AT, adenine and thymine, there's only two. There's a hydrogen bond here, and there's a hydrogen bond there. The way that these structures are made, we get a short little structure, and we get the bigger, fatter structure. They are actually so well positioned in geometry that they snap together like a puzzle piece. And this puzzle piece is going to be held together by a little bit of glue. And that glue are hydrogen bonds. So for every AT pairing, we will get two hydrogen bonds that they will share. In the case of guanine and cytosine, there's more. We still get the short piece. We still get the longer piece. And they still fit together like a puzzle piece. But the way that these are made, the GC is a little bit stronger. We have a hydrogen bond that happens here. H on an N, O, or F attached to another N, O, or F. There you go. We have a hydrogen bond that's here. H attached to an N, O, or F that's attached to another N, O, or F. And finally, we have hydrogen bond number 3. So the GC stacking is a little bit stronger. It's a little bit harder to pull apart. And the reason is because they share multiple hydrogen bonds, 3 here versus 2 from the AT pairing. Also, something I want you to keep in mind. The more hydrogen bonds, the more stable the molecule becomes. That's going to play a very important role a little bit later on when we build up to the DNA structure. Again, we're starting small. We're looking at how the pieces are fitting together, and we're going to slowly build up and up and up from that concept. All right, so this is where I'm going to stop. And then in the next video, we'll continue on and we'll talk about the pairing, and we'll get bigger and bigger as we go.